Welcome everybody to part 2 of DNA and hybridization, where we will continue talking about how hybridization is useful in real-world scenarios. Right, so the first sentence here that we have is complementarity and hybridization is the heart of many nucleic acid-based techniques. So what examples is hybridization utilized in real-world scenarios? So some examples for you could be chromosome painting, microarrays, which will be a topic later on, which we will cover, exon capture, DNA sequencing by Sanger or next generation sequencing. So DNA sequencing, either using Sanger or next gen. PCR and its various forms, as well as recombinant DNA techniques such as cloning. So we'll just add PCR here to the mix. So they all rely upon the specificity of complementary and avoidance of mismatches by using stringency and TM of the duplex covered in part 1. Right. Nucleic acid hybridization technique. So nucleic acid and hybridization, they come in hand, they work together simultaneously. And by this, nucleic acid hybridization is a technique crucial for identifying the presence of nucleic acids with specific base sequences. By utilizing this method, we can selectively capture these specific sequences, enabling us not to only detect their presence, but also quantify their abundance in a complex mixture. In biomedical applications, this technique serves as a powerful tool for targeted genetic analysis, offering insights into the composition and quantity of specific nucleic acid sequences. So, to break this down for you guys, nucleic acid hybridization techniques, it's useful to identify the presence. So identify the presence of nucleic acids, NA, with specific, keyword specific, sequences. Right. So this method selectively captures these specific sequences, enabling us to not only detect their presence, but to also quantify the abundance in a complex mixture. So by identifying these specific sequences, we're able to detect their presence, and also identify or quantify how much the abundance is within a complex mixture. Quantify the abundance in a complex mixture. Right, so that is essentially what nucleic hybridization techniques are, to identify the presence of specific sequences, detect their presence, and quantify the amount there is in a complex mixture. Right. Hybridization uses the ability of nucleic acids to form specific duplexes. Right, so when making duplexes, hybridization harnesses the unique ability of nucleic acids to form specific duplexes by exploiting the complementarity and hybridization of labeled nucleic acids, often referred to as probes. This technique allows us to pinpoint and analyze specific genetic sequences. This powerful method is foundational in molecular biology, aiding in the precise identification and study of genetic material. Right, so when making specific duplexes, hybridization labels these nucleic acids as probes. So hybridization harnesses the unique ability of nucleic acids, so binds with nucleic acids to form specific duplexes. And to identify these specific duplexes, what we do is we label these nucleic acids. So we label them, and we refer to these labeled nucleic acid forming a specific duplex. So if we combine these two, label and specific duplex, we refer to them as probes. And this technique allows us to pinpoint and analyze specific genetic sequences. So to analyze specific genetic sequences, which is what we want. And again, this powerful method is foundational in molecular biology, again, to identify and study genetic material. And as a small kind of like example here, this will be a gene. Let me label these exon gene X. Exon gene X. And here we have a unique sequence found in exon 1. Unique sequence found in exon 1. Of X. And this hybridizes to a probe of X. To a probe of X. So here we have a unique sequence of like a random exon, and this is what we want to find. And when we do the hybridization technique, this specific nucleic acid sequence is then copied to a identical copy here, and we call this probe X. Probe X. And so we can identify what we're looking at based on the original sequence. And this can be the same for something else. So if we have another sequence, and let's label this exon gene Y. Exon gene Y. We have a unique sequence here. Unique sequence found in exon 2. Exon 2. And this hybridizes to a probe of Y. Probe Y. So it's pretty straightforward. Whatever our nucleic acids hybridizes to, we can always trace it back to its original sort of coding sequence. So either gene X or gene Y. So people may be asking, okay, so we know that probes are used to identify specific exon genes, but what is a probe? So to zoom in further, usually a probe is composed of single-stranded DNA or RNA molecules. Right. Also, it's typically around, so there's a range of its length, typically a range of 20 to 1,000 base lengths, bases in length. And this is obviously labeled with a fluorescent or luminescent molecule. Labeled with a fluorescent or luminescent molecule. Right. And in some techniques, thousands or millions of probes are used simultaneously. And carry on going further. So common targeted hybridization based techniques. So we already went through it in the beginning, but some common ones that we normally use in the labs are in situ hybridization, chromosome paintings of chromosome spreads. So yeah, chromosome paintings. Let's go here, chromosome paintings of chromosome spreads. Analysis of mRNA or DNA by PCR. So PCR and then analysis of either DNA or RNA, and Sanger/dideoxy sequencing. So just for simple reference, we'll just put Sanger sequencing because we all know that. If not, there'll be a video coming out for Sanger sequencing specifically. These techniques, however, there is something that these techniques have in common. And the reason why I said maybe we use this in labs or at a small scale is that these techniques are not very scalable and detect very few genes at a time and a small number of samples. So just as a note here, these techniques specifically are not scalable. Good. Cool. However, if we were to look for something a bit more scalable, we would look at genome-wide techniques. Frequently, we want to analyze thousands of segments of DNA or RNA in each sample. So hybridization has been employed in high-throughput techniques, and these high-throughput techniques are the following: microarrays, sequence capture, example, exon capture, and next generation sequencing. Leave it like that. So microarray sequence capture, example, exon capture, and next generation sequencing are often used as genome-wide techniques, as they are able to analyze many thousands of segments of DNA, whereas the rest, such as in chromosome painting or in situ hybridization, are not able to detect thousands of segments. So those are the main difference between these techniques. And finally, moving on, just to understand briefly what two of these techniques do, mostly in universities, specifically biomed students, we would normally look at microarrays. We'll be making a video of microarrays soon, and next generation sequencing. So just to give a brief overview of how do microarrays work. So how do microarrays work? So an ordered assembly of nucleic acid probes. So ordered assembly of nucleic acid probes. And these probes are fixed in a solid surface. So keyword here are fixed in a solid surface. And the sample of interest is hybridized. So sample of interest is hybridized. Right. So just to go over it again, microarrays are ordered assembly of nucleic acid probes, so they're ordered, and the probes are fixed in a solid surface. So again, remember, solid surface, and the sample of interest, so the sample or the exon or exon X or exon Y, for example, the sample that we want to kind of expand is hybridized. Right, so quite straightforward. And lastly, what is next generation sequencing? We'll go into further detail of both of these techniques later on, but this is just a brief understanding of what these techniques do. next generation sequencing. And just to put it simply, it's a parallel sequencing of millions of molecules captured on a surface by hybridization. So parallel sequencing of millions of molecules captured on a surface of by hybridization. Done. Simple. Okay, so the summary, which I will leave it on the description bar below the video. Summary, we've defined what we mean by a probe and explained their use in hybridization. We listed a wide range of techniques that use hybridization from chromosome pasting to qPCR. We introduced high throughput techniques like microarrays, exon capture, and next generation sequencing as a scalable technique that have hybridization at their heart. So that concludes both parts of complementarity and hybridization, and we'll be moving on further on with our genomics playlist. So thank you guys. Leave a thumbs up if you like the video. If you have any questions, leave it down in the comments. Subscribe if you find this useful. And again, take care of yourselves and of each